In 2014, a geneticist at Imperial College London ran a genomic analysis on Somali DNA. He expected to find Arab ancestry, maybe Egyptian, maybe Levantine. That was the accepted story. Somali people came from Arab migrants who crossed from Yemen around 3,000 years ago. Every history book said so. Every scholar agreed. The DNA said something completely different. The results showed a genetic signature that did not match Arabia, did not match Egypt, did not match any currently living population outside of Africa. It was something older, something that had no name, something that had been sitting inside Somali cells for 23,000 years, completely invisible, until that moment. That geneticist was Dr. Jason Hodgson. His co-researcher was Dr. Connie Mulligan from the University of Florida, USA. Together, they published their findings in PLOS Genetics, one of the most respected scientific journals on the planet. The paper is titled Early Back to Africa Migration into the Horn of Africa, published June 2014. What they found did not just challenge the mainstream story about Somali ancestry, it destroyed it. And by the end of this video, you will understand why scientists are calling the Somali genome one of the most unique genetic profiles ever recorded in human history. Right now, drop your country in the comments. Where are you watching from? And tell me, have you ever taken a DNA test? What did your results say? Because what you are about to hear might completely change how you understand where human beings actually come from. Let's get into it. For decades, the official explanation of Somali ancestry was simple. Arab traders came from Yemen. They arrived on the Somali coast. They settled. Their descendants became the Somali people. Migration happened roughly 3,000 years ago. End of story. It was clean. It was neat. And according to genomic science, it was wrong. Here is the proof. There is a genetic mutation called the lactase persistence allele. It appeared in Arabian populations approximately 4,000 years ago. Today, over 50% of people in Arabia carry it. It is one of the clearest genetic markers of Arab ancestry in any population. If Somali people descended primarily from Arab migrants 3,000 years ago, that allele should be everywhere in Somali DNA. Dr. Hodgson and Dr. Mulligan measured it. Somali populations carry it at just 1.6%. 1.6%. That is not the number of a population descended from Arab migrants. That is the number of a population that had almost zero genetic input from Arabia, at least in terms of the dominant ancestral component. Their paper states directly, and I'm quoting the published text, the genetic composition of non-African ancestry in the Horn of Africa is distinct from the genetic composition of current populations in North Africa and the Middle East. Distinct. Their word, not mine. So if Somali ancestry did not come from Arabia 3,000 years ago, then where did it come from? The answer involves a date that most people cannot even process, and a direction of migration that no mainstream history class has ever taught. The dominant genetic component in Somali DNA has a name. Researchers call it the Ethio Somali ancestry component. According to the Hodgson and Mulligan study, this component diverged from all other non-African ancestries by at least 23,000 years ago. That date was calculated using roll-off and Alder statistical modeling, two of the most rigorous methods in population genomics, at least 23,000 years ago. To put that in perspective, the pyramids of Giza are 4,500 years old. The Roman Empire is 2,000 years old. Islam began 1,400 years ago. All of those combined do not come close to 23,000 years. This component predates agriculture, predates writing, predates every civilization that history has ever recorded. Dr. Orion Rayaum, assistant professor of anthropology at Lehman College, City University of New York, and co-author on related research, described this as a substantial ancient migration, not a small drift of people, a real movement of a real population, arriving in the Horn of Africa and leaving a genetic signature so strong that it still dominates the Somali genome today. Now, here is the part that makes geneticists uncomfortable. This component is classified as non-African in terms of its divergence point, meaning the ancestral population left Africa at some point, evolved in isolation outside the continent, and then returned to the Horn of Africa before 23,000 years ago, a back-to-Africa migration, a reverse journey, one that left almost no trace in any historical record, only in DNA. Dr. Hodgson confirmed in the study that this migration predates the Arabian lactase persistence allele by at least 19,000 years. And the people who brought this ancestry to the Horn of Africa arrived long before Arab civilization existed in any form. And for those wondering, does this contradict Islamic genealogy? No. The Quran in Surah Al-Hujurat, chapter 49, verse 13, states that Allah created human beings into nations and tribes so that they may know one another. These ancient genetic layers do not erase Islamic lineage. Islamic genealogy tracks ancestry through names, faith, and community. Genetics tracks the biology of cells going back 23,000 years. Both records exist. Both are true. They are measuring different things. What genetics is showing us is that the foundation of Somali ancestry is far older and far more extraordinary than any recorded history captured. When this ancient population arrived in the Horn of Africa, the land was not empty. It was already home to hunter-gatherer groups, ancient lineages, people who had lived in that specific land for tens of thousands of years in near total isolation, some of the deepest-rooted human populations on the planet. In 2020, a study published in Science Advances titled Ancient Genomes revealed complex patterns of population movement, intermixture, and selection in the Horn of Africa, sequenced ancient DNA from multiple burial sites across East Africa. What the bones showed was that the arriving pastoralist populations and the existing ancient forager groups did not fight to extinction. They mixed. One individual's skeleton would carry 50% ancient forager DNA and 50% pastoralist DNA at the same time. The same body, the same bones, two completely different worlds inside one person. This merging, over centuries, created the ancestral genetic foundation of the Cushitic populations, the language family that Somali belongs to, and the cultural and biological root of the Somali people. But here is what most documentaries skip over entirely. The ancient forager layer, the people who were already in the Horn of Africa when the Back to Africa migrants arrived, they have no name, no recorded language, no archaeological signature that has been fully identified. They vanished into the merged population, leaving only their genetic material behind. Their DNA is inside living Somali people today, and it has been there for thousands of years. And until ancient DNA science was developed, nobody even knew they had existed. This is the real foundation, not Arab traders from 3,000 years ago. A deep, layered, complex genetic history going back tens of thousands of years, built from multiple ancient populations that merged in one of the most significant human mixing events in prehistoric Africa. And sitting on top of all of that, a Y chromosome marker so dominant in Somali men that when geneticists traced it, they found it in populations that had no direct historical contact with Somalia at all. In Somali men, the dominant Y chromosome haplogroup is E1b1b, specifically the subclade E-V32. According to the Hodgson study and Y chromosome data, published in the European Journal of Human Genetics by Sanchez and colleagues in 2005, E1b1b is the defining paternal marker of Horn of Africa Cushitic populations. Dr. Fulvio Cruciani of Sapienza University of Rome, Italy, who published multiple peer-reviewed papers on E1b1b across Africa and the Mediterranean, established that the parent lineage EM78 originated in Northeast Africa approximately 19,000 years ago, the Horn of Africa, Somalia, and Ethiopia. That is the birthplace of this haplogroup. From there, it traveled. Over thousands of years, E1b1b spread into North Africa, into the Sahara, into the Mediterranean, into Europe. Today, it appears in Berber populations across Algeria and Morocco, in ancient Egyptian populations, in Albanian and Greek and Italian men. The highest genetic diversity of E1b1b, which in population genetics marks the place of origin, is in Northeast Africa, not Arabia, not the Levant. In Northeast Africa, the haplogroup did not arrive in Somalia from outside. It was born there, and it moved outward from there to populations that later built some of the most famous civilizations in history. In 2024, Amal Abshir Mohammed, researcher at the Bureau of Forensic Science in Garowe, Somalia, published a forensic Y chromosome study analyzing Somali males across Mogadishu, Garowe, Bosaso, and Galkayo. The results confirmed what the genetic databases had shown: a consistent, homogeneous paternal profile across all 4 cities. One shared ancient paternal origin, present and measurable in living Somali men today. That is the father's line. The mother's line is different. And what Somali maternal DNA reveals is something that most people, even most scientists, find difficult to fully absorb when they first hear it. Mitochondrial DNA passes only through mothers. It does not mix. It does not change the way autosomal DNA does. It is a direct, clean line from mother to daughter going back thousands of years. The dominant mitochondrial haplogroup in Somali women is L3. L3 is the maternal ancestor of every non-African person alive on Earth today. Every European, every Asian, every Native American, every human being whose ancestors left Africa at any point in history. Their maternal lineage traces back through L3. Without L3, the entire non-African human family tree does not exist. The deepest, see, most ancient branches of L3, the versions that indicate geographic origin, are found in East Africa, in the Horn of Africa, in the same region where Somali women have lived for thousands of years. This is not a distant connection. This is origin. According to mtDNA frequency data from anthropological genetics research, over 60% of Somali maternal ancestry belongs to the L haplogroup family, with L3 as the dominant branch. These smaller percentages carry M1 and HV1, markers that appear in North African and Near Eastern populations. But these are traces of ancient coastal contact, not population replacement. The core maternal lineage of Somali women is one of the oldest human maternal lineages on the planet. It did not come from outside. It has been in that land since before the rest of the world's family trees even started branching. And now, here is where the data gets uncomfortable. Inside the Somali genome, there is a genetic signal that tells scientists something dramatic happened to this population at some point in deep history. A signal that says this population nearly disappeared. And the evidence for it was found not in ancient bones, not in archaeological digs, but in the immune system of living Somali people in 2020. In 2020, Dr. Hanna Olila and a team at the Finnish Institute for Molecular Medicine, University of Helsinki, Finland, published a study in Scientific Reports. See to title, Genome-wide analyses disclose the distinctive HLA architecture and unique origin of the Somali population. They analyzed 900,000 genomic markers per individual across 95 Somali people. This is one of the most detailed molecular analyses ever done on a sub-Saharan African population. Their finding, the HLA system, the part of the genome controlling immune response, in Somali people is unusually homogeneous, far more uniform than in neighboring Ethiopian, Kenyan, or Yemeni populations. Dr. Alila's team wrote in the published paper, direct quote, "Ethnic Somalis are an East African population with unique HLA composition." In population genetics, this level of uniformity has one explanation: a bottleneck, a moment when the population collapsed to a very small group of survivors. When those survivors rebuilt, all future generations carried only the genetic diversity of those few people who made it through. Well, the study also found 9 pharmacogenetic variants in the Somali genome, genetic markers affecting how drugs are processed by the body, present at frequencies found in no other world population. These are almost certainly survival adaptations, mutations that became dominant because the individuals who carried them lived when others did not. The Somali genome carries the biological record of a near-extinction event. And now, inside that same extraordinary genome, something even more disturbing was found, something that should not be there, a genetic component belonging to a population that, by all logic of history and migration, should no longer exist. Scientists gave it a name. They called it a ghost. Ghost population. In genetics, a precise technical term. A population that no longer exists as a living, identifiable group. They merged. They were absorbed. They vanished from the earth as a distinct people. But their genetic material survived inside the descendants of those who absorbed them. It lives on, hidden in DNA, invisible to history, visible only to modern genomic science. The Hodgson and Mulligan 2014 PLOS genetics paper identified the Ethio Somali component as exactly this: It is, in their words, found in Somali and Ethiopian populations. Bob shows little inter-individual variance within these ethnic groups and is estimated to have diverged from all other non-African ancestries by at least 23,000 years. It does not match any living population. It is not found anywhere else at significant frequency. It is a ghost, and it makes up the majority of the Somali genome. The study's authors concluded prehistoric hunter-gatherer populations were much more dynamic than usually assumed. Dynamic is a careful scientific word. It's what it means in direct terms. The ancient people of the Horn of Africa were doing things, going places, and building genetic histories that modern science is only now beginning to understand. And the mainstream story that was taught for generations was not even in the right century. Now, here is the key question that the ghost component raises. And if this lineage is found almost exclusively in Somali and Ethiopian populations, how did traces of related ancestry end up in populations along the Indian Ocean, thousands of miles from the Horn of Africa? In communities that have no recorded historical connection to Somalia? The answer is a 2,000-year-old trade network, and the people running it were Somali. The port of Zayla, on the Somali coast of the Gulf of Aden, is referenced in ancient Greek geographical texts written over 2,000 years ago under the name Avalites. Roman-era merchants mentioned it. Indian trading records referenced it. Archaeological excavations at ancient Somali coastal sites have found Indian ceramics, Persian glass beads, and Chinese coins in the same archaeological layers. 3 different civilizations, separated by thousands of miles, all present in one location. Where Professor David Reich of Harvard Medical School, USA, and Dr. Pontus Skoglund of the Francis Crick Institute, UK, whose teams have produced some of the most comprehensive ancient African DNA research ever published, confirmed through large-scale genomic studies between 2017 and 2020 that coastal East African populations show measurable genetic exchange with populations from across the Indian Ocean. And critically, those genetic signals follow the known maritime trade routes almost exactly. The people running these ports were not passive traders receiving goods from outside. The genetic evidence shows they were the source. The genetic signals travel outward from the Horn of Africa, not inward to it. This is the part no Western history textbook covers. But even within Somalia, there is a split in the data. A divide in the genome that reveals 2 completely different histories existing in the same country at the same time. And that divide is the final piece of this story. Dr. Mayaolila's 2020 University of Helsinki study found something critical inside the Somali genomic data. Coastal Somali populations, the people living near ancient port sites, carry measurably higher levels of West Eurasian and Arabian genetic admixture than inland populations. The maritime contact zones absorbed outside DNA. The coast was open to the world. But the inland populations, the pastoralists living far from the coast, carry almost none of that outside admixture. What they carry instead is the purest surviving expression of the Ethio Somali ghost component, the 23,000-year-old lineage, almost completely undiluted. 2 different genetic profiles, 1 country, existing side by side for thousands of years. One Somalia that was connected to the ancient world, trading, absorbing, exchanging with Arabia and Persia and India. And one Somalia that preserved something ancient beyond all of that. Something that was old before those civilizations existed. And then, in 2024, a pangenomic study from the University of Helsinki, using advanced genomic variation graph analysis, found that previous research had been underestimating the depth of Somali genetic uniqueness. The standard tools used in population genetics were not precise enough. The real divergence of the Somali genome from all other world populations, the real age of its uniqueness, is likely even greater than the numbers we have cited in this video. Greater, deeper. So it's more ancient than 23,000 years. 25 million people alive today carrying a genetic signature that predates every recorded civilization, that survived a near extinction, that spread its paternal marker to 3 continents, that holds the maternal ancestor of every non-African person on Earth. That was built from the merging of ancient worlds whose names no one remembers. And still, there are questions the science has not answered. So in the Ogaden region, deep in the Somali interior, archaeologists have found human remains that do not match Cushitic ancestry, do not match the ancient forager lineage, and do not match any population discussed in this entire video. A 3rd genetic profile. sitting between all the known categories, or completely outside all of them. Who were they? How long were they there? And what does their DNA tell us about a chapter of human prehistory that has never been written? That is the next video. So subscribe and turn on notifications because that answer is coming. And based on everything you just heard, you already know it is not going to be what anyone expects.